In the previous video, we discussed 5' capping, the first important modification of pre-mRNA. But capping alone is not enough. The RNA still contains unwanted regions that need to be removed before it becomes functional. That process is called splicing. When a gene is copied from DNA into RNA, the cell first forms pre-mRNA. This pre-mRNA contains useful regions called exons and unwanted regions called introns. Splicing removes the introns and joins the exons together to form mature mRNA. You can think of it like editing a recorded video. Before uploading, you cut out unnecessary clips and keep only the important parts. Cells do something very similar with RNA. This process is carried out by a complex called the spliceosome, which is made of proteins and small nuclear RNAs, or snRNAs. Now let's understand the different types of splicing. The first type is constitutive splicing. This is the default and most common form of splicing in eukaryotic cells. Here, the spliceosome removes introns in the exact same pattern every single time. That means the same exons are always joined together, so the final mature mRNA remains the same. In this type, the spliceosome works normally. It recognizes the splice sites on both sides of an intron, forms the lariat structure, cuts the intron out, and joins the neighboring exons together. The second type is alternative splicing. This is one of the most important reasons why humans can produce so many different proteins from a limited number of genes. Here, the spliceosome is still used, but unlike constitutive splicing, it does not always choose the same splice sites. Instead, the spliceosome can skip certain exons, include specific exons, or cut at different positions. Because of this, the same pre-mRNA can produce multiple different mature mRNAs. For example, one cell may produce this by skipping only introns, while another cell may skip all the introns and some exons as well, like exon 3 in this case. So this type creates different proteins from the same gene. You can think of it like making different sandwiches using the same bread, but changing the fillings each time. This type of splicing is extremely common in humans, especially in brain cells, muscle cells, and immune cells, and it plays a huge role in development and cell specialization. The third type is self-splicing. This type is unusual because no spliceosome is used at all. Instead, the RNA removes its own introns by folding into a special three-dimensional structure. The RNA itself performs the cutting and joining reaction. That means the RNA acts almost like an enzyme. RNAs that can catalyze reactions like this are called ribozymes. This is very different from normal spliceosome-mediated splicing because here, the RNA is both the molecule being processed and the molecule performing the reaction. Self-splicing is mainly found in some microorganisms, mitochondria, chloroplasts, and other primitive cellular systems. The last type is trans-splicing. Normally, in regular splicing, exons from the same RNA molecule are joined together. But trans-splicing is unusual because exons from two different RNA molecules can be joined together. In many cases, this process still uses a spliceosome-like mechanism. The difference is that instead of connecting exons from one transcript, the spliceosome joins exons coming from separate RNAs. This type of splicing is seen in some protozoa, trypanosomes, nematodes, and a few specialized systems.